My name is Joshua Mugabi. I welcome you to Topic 2, Nutrition in Plants. Our learning outcome A. Evaluate the relationship between the structure of chloroplast and photosynthesis in C3 and C4 plants. Details of biochemistry are not required. Our learning activity is C. In groups, learners search and discuss the specific pathways involved in the synthesis of proteins and lipids using intermediates ATP, NADPH, G3P in reactions of photosynthesis. Using computer-generated infographics, learners present their findings to the rest of the class. The role of photosynthetic intermediates in biosynthesis. Item 1. Emphasizing from photosynthesis to growth. A student observes that plants supplied with adequate light grow faster and produce more leaves than those grown in shade. The biology teacher challenges learners to explain how products of photosynthesis are used to synthesize protein and lipids. Task. Identify the photosynthetic intermediates involved in biosynthesis. State the origin of ATP, NADPH, and G3P in photosynthesis. Explain how these intermediates contribute to protein synthesis. Identify the photosynthetic intermediates involved in biosynthesis. We have ATP, NADPH, and glyceraldehyde 3-phosphate. State the origin of ATP, NADPH, and G3P in photosynthesis. ATP and NADPH are produced in the light-dependent stage. G3P is produced in the light-independent stage, Calvin cycle. Explain how these intermediates contribute to protein synthesis. G3P is converted into glucose and other carbohydrates. Carbohydrates provide carbon skeleton for amino acid synthesis. ATP supplies energy for active transport of nitrates and peptide bond formation. NADPH provides reducing power for conversion of nitrates to amino acids. Let's go now to the role of photosynthetic intermediates in biosynthesis. Item 2. Emphasizing of protein synthesis pathway in green plants. During a practical lesson, learners trace the pathway from carbon dioxide fixation to formation of plant proteins. Task: outline the pathway linking photosynthesis to protein synthesis. State the role of ATP in this pathway. State the role of NADPH in nitrogen assimilation. Outline the pathway linking photosynthesis to protein synthesis. Carbon dioxide is fixed during the Calvin cycle. G3P is formed. G3P forms glucose and other carbohydrates. Carbohydrates are converted into organic acids. Organic acids combine with nitrogen to form amino acids. Amino acids join to form proteins. State the role of ATP in this pathway. ATP provides energy for nitrate uptake from soil, reduction of nitrates, formation of peptide bonds during protein synthesis. State the role of NADPH in nitrogen assimilation. NADPH reduces nitrates to nitrites, nitrites to ammonia. Ammonia combines with carbon skeletons to form amino acids. Still on the role of photosynthetic intermediates in biosynthesis. Item 3. Lipid synthesis in chloroplasts. Students examine oil-rich seeds such as sunflower and groundnuts and are asked to relate lipid formation to photosynthesis. Task: identify the photosynthetic intermediate used directly in lipid synthesis. Explain how G3P contributes to lipid formation. State the role of ATP and NADPH in lipid synthesis. Identify the photosynthetic intermediates used directly in lipid synthesis. We have glyceraldehyde 3-phosphate, G3P, explain how G3P contributes to lipid formation. G3P is converted to glycol. Glycol combines with fatty acids to form triglycerides, the lipids. State the role of ATP and NADPH in lipid synthesis. ATP supplies energy for fatty acid synthesis. NADPH provides hydrogen for reaction during fatty acid formation. Still under role of photosynthetic intermediate in biosynthesis, item 4, emphasizing on the effect of reduced light intensity. Plants grown under low light show reduced protein and lipid synthesis. Task: explain the effect of how low light on ATP and NADPH production. Relate this effect to reduced synthesis of proteins and lipids. Predict the overall effect on plant growth. Explain the effect of low light on ATP and NADPH production. Reduced light lowers the rate of light-dependent reactions. Less ATP and NADPH are produced. Relate this effect to reduced synthesis of protein and lipids. Reduced ATP limits energy requiring biosynthetic reactions. Reduced NADPH limits reduction of nitrates and fatty acid synthesis. Less G3P is formed, reducing carbon skeletons. Predict the overall effect on plant growth. Slower growth, poor leaf development, and reduced biomass accumulation. Still on the role of photosynthetic intermediate in biosynthesis, item 5. Looking at the pathway analysis. Students are required to draw a flow diagram showing the relationship between photosynthesis and biosynthesis. Task: construct a flowchart linking photosynthesis to protein and lipid synthesis. Label the role of ATP, NADPH, and G3P. Construct a flowchart linking photosynthesis to protein and lipid synthesis. Light-dependent stage produces ATP and NADPH. Calvin cycle gives G3P. G3P is converted to glucose, which produces carbon skeleton. ATP and NADPH are used in amino acid synthesis, which produces protein. G3P is converted to glycerol and fatty acid to produce lipids. Label the role of ATP, NADPH, and G3P. ATP provides energy required for biosynthetic reactions. NADPH reduces nitrates and fatty acid synthesis. G3P used in the formation of carbon skeleton. Marking emphasis. Correctly identify intermediates, ATP, NADPH, and G3P. Explain their specific roles in protein and lipid synthesis. Show clear linkage between photosynthesis and metabolism. Use correct biological terminology. Apply knowledge to real plant growth scenarios. Conclusion. Photosynthesis provides not only carbohydrates but also the energy, reducing power, and carbon skeletons required for synthesis of proteins and lipids, demonstrating its central role in plant metabolism. Thank you for participating in Topic 2, Nutrition in Plants. Our learning outcome A. Evaluate the relationship between the structure of chloroplast and photosynthesis in C3 and C4 plants. Details of biochemistry are not required. We have been looking at learning activity C. In groups, learners search and discuss the specific pathway involved in the synthesis of proteins and lipids using intermediate, ATP, NADPH, G3P of reactions of photosynthesis. Using computer-generated infographic, learners present their findings to the rest of the class. If this has been useful to you, visit me at Mugabi Joshua Biology on YouTube.